will wait for another 2 to 3 minutes. Then we will start. So, okay, we'll start our class. I'm sharing my screen. Screen is visible.
Yes, sir. Yes, sir.
Yes, sir.
Okay, we'll discuss about our unit 3, that is cell cycle. Uh, then what is cell cycle? The cell cycle is a series of events that a cell goes through as it grows and divides in cell division, where a parent cell split into two daughter cells, and this cycle of cell division includes different phases of growth, DNA replication, and ultimately cell division. If you go to the this figure, the cell cycle is a sequence of events. That is, first one is cell growth, then the DNA replication, then preparation for mitosis, then cell division. Cell division occurs in mitosis process. And the if you go to the different pages of this cell cycle, if you go to the interpage, this is called interpage. This interpage is G1, S and G2 page. If you go to the G1 page, that is here in this figure, that is cell growth page, and this in this page cell grows and carries out in normal function. Here the after the cell division, cell will grow and carries out its normal function. After the G1 page, then the cell enters into the S page. S page means synthesis page. In this S page, the DNA replication occurs, means the DNA copy number increases from 1 to 2 because one cell is divided into two cells. So the DNA copy number also uh, copied from one cell to two cells, two daughter cells. So in this case, DNA replication occurs and resulting to into identical copies of each chromosome. Here, the DNA replication occurs to form a identical copies of each chromosome. Then in the G2 page is the preparation for mitosis, and in this phase, the cell prepares for mitosis, ensuring that DNA replication is complete and the cell is ready to divide. In this phase, after the DNA replication, in this phase, DNA replication will be complete and ready to divide, and it go for the cell division phase, that is M phase or mitosis phase. In the mitosis phase, you will see that in the mitosis process, the nuclear division occurs where the duplicated chromosome are separated into two identical sets. After the two identical copy numbers, then the cell divides into two identical sets to form a two daughter cell. In the mitosis process, there is a five phases. One is karyokinesis and another one is cytokinesis. In karyokinesis process, that is a prophase, metaphase, anaphase and telophase. In cytokinesis process, the cytoplasm divides finally and give rise to two separate daughter cells. Then if you go to the in detail of mitosis process, in mitosis, this process of cell, in this process, cell duplicate into two daughter cell, two diploid daughter cell, or reproduction during which one cell gives rise to two genetically identically daughter cell. Strictly applied, the term mitosis is used to describe the duplication and distribution of chromosome. Here, the duplication and distribution of chromosome. If here the chromosome number is 46, here then also the chromosome number is 46, and here also 46. And here the duplication of the chromosome occurs, identical duplication, then two diploid cells are produced. But in meiosis is a specialized type of cell division that reduce the chromosome to half. Half means this is if this is the 46 number of chromosome, after meiosis 1 or meiosis 2, the number of chromosome will be 23, 23, 23. But here the four uh, daughter cell will produce having half number of chromosomes. If we go to the basic difference between the mitosis and meiosis, in mitotic division, a single division result into two daughter cell. A single division result into the two daughter cell. But in meiotic division, here two meiosis one and meiosis two occurs. These division result into four daughter cell. Here two daughter cell. Here four daughter cell. In the mitosis is known as equational division, equal number of division, and this because the daughter cells have the same diploid number of chromosomes. Suppose here the chromosome, if the division or replication start, here the number of chromosome 46. In the two diploid cells, the number of chromosome also here 46 and here also 46. And the cells have the same diploid number of chromosome as the parent. Uh, this is the same as the parent, the chromosome number. But in meiosis 1, known as the reductional division, here the reductional division occurs. This is because of the chromosome number is reduced to half. because from 46 the number is reduced to 23 and 23, and this 23 is copied into 2 23 23 and 23 23. Here we found 4 daughter nuclei. In meiosis 2 is known as equational division. The meiosis 2 is also called mitosis of the meiosis 1. uh, from the this meiosis 1, the copy number is 2 2 from 1 daughter nuclei to 2 daughter nuclei. Here 1 daughter nuclei to 2 daughter nuclei. Total the daughter nuclei is 4 in meiosis process. If you go to the pro-page of the mitosis, the pro-page is short and doesn't comprise any phase. In the pro-phase, there is no any phase is occurring in this mitosis process, but in the pro-phase of the meiosis, that is different 5 phases, that is leptotin, zygotin, pachitin, diprotin, and diakinesis process. So there is no pairing of the chromosome because here the chromosome is just copied from the parent cells, but here the but here the chromosome number is reduced, and here in mitosis the crossing over or chiasma formation during the prophase will not occur, but in meiosis 1 of prophase 1, the zygote in the zygote stage of the prophase, the pairing of the chromosome occurs during the pachytid, and the crossing over also occurs, and the chiasmata also are formed in diploid stage. In meiosis, uh, the synaptonymal complex is formed during the zygotic stage of the prophase one, but in mitosis, the synaptonymal complex will not form. And in anaphase, involves the separation of the chromatids of each chromosome in the mitosis process. But in meiosis, here the anaphase is two in anaphase one and anaphase two. During the anaphase one, homologous chromosomes are separated while the chromatids remain attached to their centromere, but in anaphase 2, the chromatids are separated, resulting splitting into 4 daughter nuclei. If we go to the in detail of the prophase of the mitosis, in the prophase of the mitosis, chromosome condense and become visible, and spindle fibers emerge from the centrosome. From this centrosome, the spindle fiber has emerged, and nuclear envelope breaks down. The nuclear envelope breaks down because DNA will be condensed, and the chromosome will be copied, and this centrosome move towards the opposite pole, and here the centrosome 1 is here and the centrosome 2 is here, and this centrosome will move to this side to form a opposite poles. And in prometaphase, the chromosome continue to condense. Here the chromosome will continues to condense to form a chromosome in the central axis of this spindle fiber, and the kinetochore appears at the centromeres. Here the kinetochore appears in the centromeres, and mitotic spindle, this is the mitotic spindle, the spindle microtubules attached to the kinetochore, and the mitotic sorry, the microtubules are attached to this kinetochore, and it holds the chromosome at the center. And in this metaphase, the chromosome are lined up at the metaphase plate, that is in the center, correct center. Then each sister chromatid is attached to the spindle fiber originating from the opposite pole. These are attached to the mm, different folds of these chromatids. And in anaphase, the separation of this in anaphase, centromeres are split into two, and this is one centromere, and another one is uh, one centromere, and the sister chromatids are now called chromosome. These are two sister chromatids, and these are called one chromosome, and this one chromosome then divides in telophase to form 2 daughter nuclei, and this chromosome arrives at opposite poles of these mm, uh, centromeres, and nuclear envelope are formed over these daughter nuclei, and the mitotic spindle also break down because here the cell will be separated into two parts, and the spindle fibers continues to push poles apart, and the spindle fiber continues to pulling of these two daughter nuclei. And then after the karyokinesis means division of this nucleus, then cytokinesis occurs. In cytokinesis process, uh, the cleavage of furo separates the daughter nuclei. This is called contractile ring. In plant cell, a cell plate or precursor to a new cell wall separates the daughter nuclei. But here in animal cell, the contractile ring will form. After this contractile ring, it will separated into two daughter nuclei. So this is the mitosis of the animal cell. Then if we go to the meiotic event in meiosis, that is this is meiosis one and this is called meiosis two. In meiosis one, one is interphase and another is one is prophase one, prometaphase one, metaphase one, anaphase one, telophase, and cytogenesis. Now, in the interphase process, the chromosomes are duplicated during the interphase, and the resulting sister chromatids are held together at this centromere, and the centrosome are also duplicated. Here, the centrosome also duplicated like this one centrosome and another one centrosome. This prophase one is lengthy process because this prophase one is 5 steps. In the prophase one, the chromosomes are condensed and the nuclear envelope was fragmented because if nuclear envelope will not fragmented, then the condensation or any division will not occur. Then homologous chromosomes bind firmly together along their length in this figure, and the forming a tetrad because there is non-homologous homologous chromosome form a tetrad, and this tetrad will form a chizomata between non-sister chromatid. Non-sister chromatid means two different chromosomes. Crossing over occurs at this chizomata, and the spindle fibers emerge from the centromere. After the this process of this chizomata formation and tetrad formation, the spindle fibers also arises because this cell will be separated after this prometaphase, metaphase, and anaphase. In the prometaphase, this homologous chromosome after the chizomata formation and the separation of this homologous chromosome are attached to these spindle fibers, and these spindle microtubules are the fused kinetochore and shared by the sister chromatid. Then the chromosome continue to condense after the chizomata formation. Then the chromosome continue to condense, and the nuclear envelope completely disappears in prometaphase. And in metaphase, the same as mitosis metaphase, the homologous chromosome randomly assembled at the metaphagic plate that is in the center of the cells, and where they have uh, attached to the different microtubules. And in anaphase, the spindle microtubules pulls the homologous chromosome apart, and the sister chromatid are still attached to the centromere. Here, the sister chromatid are attached till the centromere in anaphase. The end, the pull the sister chromatid to the end of this shell. And in telophase and the cytokinesis, the sister chromatid arrives at the opposite pole, each pole, and the cell and begin to decondense. Here the cell is begin to decondense, and a nuclear envelope is formed around the each nucleus. Here the one nuclear envelope will form. Here also one nuclear envelope will form to separate these two different sister chromatids. And the cytoplasm is divided by a cleavage or furo. This is by the contractile ring, and this result into two haploid cells. That is, here if the number is 46 and it will reduce to 23 and 23 each, and each cell contains one duplicated copy of each homologous chromosome pair. Here the number is 23 and also here the number is 23. Sorry. In meiosis 2, that is a mitosis process of this meiosis 1, and here the sister chromatids condense and a new spindle begins to form, and the nuclear envelope starts to fragment. Here the nucleus envelope are forming this anaphase and telophase phase, but in prophase 2 the nuclear envelope also starts to fragment, and in prometaphase the nuclear envelope disappears and the spindle fiber engage individually at coordinator on the sister chromatids, and the sister chromatid line up at metaphase plate at the metaphase process, and these sister chromatids are pulled apart by the shortening of the kinetochore microtubules, and non-kinetochore microtubule lengthen the cells because they will pull the sister chromatids apart from the centromeric plate, and it will then the chromosome arrives at each pole. Here is one pole, here is the one pole, and this nuclear envelopes around four nuclei. That is here one, this is two, this is three, and this is four. And this four involves around four nuclei, and this cleavage bureau is formed during the cytokinesis process to give two daughter haplo, two, two cells into four daughter haploid cells. Here from parent cell to two, sorry, parent cell to four daughter haploid cell. That is, if the parent cell chromosome number is 46, here the chromosome is 23, 23, 23, 23 each. This is the process called mitosis and meiosis. Till the slide understood?
Yes, sir. Yes, sir.
Okay.
Mm.
Then we'll go for the main topic, that is cell cycle regulation, how the cell cycle is regulated. As we described, the cell cycle is different pages. One is interphase and now another one is mitosis phase. In the interphase, that is a G1S and G2 phase. Here is the G0 phase, that is quiescent phase or resting state. We'll discuss about this G0 and G1S and G2 and mitosis phase. Yeah, I didn't know the cell grows and copies its DNA. In the G1 is the growth phase of the cell, and after the growth phase, the synthesis phase. Here the DNA replication occurs, and in the G2 phase, the more growth of the cell will occur and preparation for this mitosis process. And the G0 phase is the resting state where the cell performs its function and it is not prepared to divide. If we go to the G1 page, during the G1 page, this G1 page we called it growth page. In this growth page, the cell will grow, cell will periodically grow and metabolically active, and all the metabolic enzymes are active in this G1 page. And in this G1 page, it will prepare for DNA replication, and replication will occur in this A space. But here the prepare all the for replication, all the process will prepare in G1 page. If this G1 page will metabolically active or not, they will check by different checkpoint, that is G1 to S checkpoints. In checkpoint, it assesses the readiness of for cell division, that is G1 to S. If the cell is ready or not, it will check by the G1 checkpoint. If error occurs, if the cell is not ready for the replication, then no progression of the S page will occur. Suppose here in G1 page, any metabolically active enzyme or the replicating enzyme are not formed or activated in this G1, then it will check and uh, stop to enter S page, and it will go for this resting page. In this resting page, all metabolically active enzyme will be activated. Then it will give signal to go for this S page. Then, if these metabolically active enzymes are not activated, then it will no progression of the S page, and cell cycle is arrest here in G1 page. If it will check no error and everything is fine here in G1 to S. Here it's if the cell know or any signal know that there is no error and everything is fine, then the progression of the S phase will occur and DNA replication will take place, and this proceed to the remaining phase of the cell cycle. Then it will go for the DNA replication phase. If 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 DNA damage is found in this G1 phase, if DNA damage is found in this G1 phase, then the uh, one protein is there. This is called tumor suppressor gene. That is P53. This is protein, and 53 is its molecular weight. We'll discuss about the. We'll elaborately discuss about this tumor suppressor gene. You know that if DNA damage will occur, it will activate your P53. Then this P53 activated CDK inhibitor. We'll discuss about this CDK inhibitor. This is cyclin dependent kinase inhibitor, and this inhibitor arrests the cyclin and CDK complex, and this inhibition of this cyclin and CDK complex will arrest the G1 to S transition of the cell, and it will arrest the and pause at cell at G1. It will pause the cell at G1. If DNA damage will not occur, then P53 is not activated, then cell cycle will go from G1 to S. If we go to the cell enters to the S phase for replication, then this is the crucial period because maximum time DNA damage or error will check out at this interface of the S stage. In S phase, this is the crucial period during interphase for DNA replication, and this prepares for cell for subsequent stages of cell division, particularly mitosis. After the replication, then it will go for the growth phase, and then mitosis will occur. Then after this, the successful transmission of genetic information to the daughter cells, and if this phase is totally tightly regulated, and this aim for accurate duplication of the DNA molecules, and this plays a significant role in maintaining fidelity because DNA or gene in the DNA gene is there. If gene is particularly or properly replicated, then it will give rise to a good mRNA and protein synthesis will occur. If mutation will occur, then it will go to different types of diseases like cancer and different types of neurological or any other diseases. So this page is very important for the cell cycle for the replication of DNA, and this plays a significant role in maintaining fidelity of this cell cycle, and this ensures fidelity and integrity of the genetic material because all the genetic material of the cell is replicated in this stage. After A stage, then the cell undergo from S to G2 phase, because in G2 phase more growth will occur and preparation for mitosis. All the enzymes or all the proteins are synthesized in this G2 phase because it will go to the mitosis process or division. And in G2 phase, the cell is also undergoes periodically growth after the DNA replication, and it will also check the DNA replication fidelity. All the replication is uh, properly occurred or not, or any mutation or damage occurred during this replication process. If this occur, then it will correct the DNA by different DNA DNA repair enzymes are there in this stage, and it will uh, give rise to a good replication process or error-free replication process. And this G2 phase also assessment the DNA damage repair, and this will If the assessment of the DNA damage repair is okay, then it will go for the M phase progression or mitosis occur and division into two daughter cells. If the surveillance and preparation, this is ensuring successful. If this is not, the DNA damages occur or any changes will occur, then the G2 phase will here arrest, and uh, there is no progression of the cell from G2 to M page. Then if we go to the key checkpoint from G1 checkpoint, if you go to the G1 checkpoint, G1 checks the DNA damage. Here also always check the DNA damage, cell size, cell is proper developed or not, or any growth signal. If growth signal will found, then the cell will undergo division or the cell is under arrest condition. Then this P53 or RB, that is retinoblastoma protein, like this, they are major regulator. They will check DNA damage occur or not. Then it will progress from G1 to SP. If you go to the G2 phase, G2 checkpoint, that is S to G2. In this S to G2 checkpoint, it ensures DNA replication is completely occur or not, or any error is there or error free. If error free is there, then it will progress from S to G2 phase. And this will also check by the P53, cyclin B and CTK1. And another one is M checkpoint, that is G2 to M checkpoint. In this G2 to M checkpoint, it ensures proper chromosome alignment before separation, because in the growth phase the proper chromosome alignment will occur properly occur or not, and this will be checked by three different proteins, that is MAT2, BOFR1 and CTC20, and this is the complete cell cycle regulation by different checkpoints. If we go to the regulator of the main regulator of the cell cycle, then the main regulator of the cell cycle of the G1 to S or S to G2 or G2 to the major regulator is the here major regulator is the cyclin and CTK complex, and it is cyclin and CTK. Please mute. These are two major regulators. This cyclin and CTK, they both are bind together to form a cyclin CTK complex, and this CTK means cyclin dependent kinase. It, this protein is totally depends on cyclin. If cyclin will not bind to the CTK, then G1 to S will not occur, or S to G2 will not occur, or G2 to M will not occur. So this CTK and cyclin complex is very very important. After the binding of the six cyclin dependent kinase and cyclin bind to the cyclin dependent kinase CTK, then it will phosphorylate, and this phosphorylation activates this cyclin CTK complex, and this cyclin CTK complex will activate binds to the target protein, and this target protein leads to the uh, progression of the G1 to S, G1 to S. Then if you go to this cyclin and CTK complex, if you go to the different phases in G0 phase, this is question phase or resting phase, the cyclin C and CTK3 is present. Cyclin C and CTK3 complex will present at G0 phase. If you go to the G1 phase, in G1 phase,
Sir, can I ask you one question?
yes,
Sir, see, what is CDK3? I do not idea about this.
this, this is a different variants of the cyclin dependent kinase. Okay, this is the enzyme. CDK3 is the enzyme, CDK4, 2, 6, CDK1. These are all enzymes. These are kinase enzymes. These are 3, 2, 6. These are variants, different variants, because of the molecular weight. If the molecular weight of the one CDK is different from the other CDK, the name will be different. Okay,
Okay.
okay.
Yes.
Okay, CTK is the main regulator of the cyclin and CTK complex is the main regulator of the cell cycle. If you go to the different phases, that is G0 phase, G1 phase, GS phase, G2 phase and M phase. The cyclin also different and the CTK active CTK also different because in this phase the CTK and cyclin this cyclin D and E binds to the this CTK 4 to 6, then this cycle is activated and will continue to G2 S phase transition. If the this CTK and cyclin binding will not occur properly, then this process is inhibited. As we discussed here, like this. If DNA damage is occur, then P53 activated. These P53 activates CDK inhibitors. This is cyclin dependent kinase inhibitor. This inhibitor binds to the this G1S cyclin CDK complex and it will pause or halt at this G1 page. If DNA is properly error free or properly replicated, then this protein will not synthesize this P53 protein. Then this cyclin and CDK complex will forward the process G1 to S phase. Then in G1S, this also this CDK and AE binds to this CDK2. Then it will forward to H to G2 phase. In G2 phase, this cyclin also forward bind to this CDK2 and CDK1 to forward from G2 to M. And in M phase, this cyclin will bind to the progression of the mitosis process and starts of the mitosis. And this is called the main or key regulator of the different checkpoints, CTK regulators. This will regulate different phases of the cell cycle. After the key regulation, then another regulator of the cell cycle is P53. We described here the P53. If we go to the this elaborately tumor suppressor gene, this is called P53. P53 suppress. What suppress? It, P53 will check DNA is mutated or error free replicated. It will occur or not. If this mutation or error is there in DNA, these P53 halt at their stage and will repair that DNA. And this is very important. And in case of DNA damage, this is if P53 can cause programmed cell death. If it check the DNA will be not properly repaired, then it will go for the apoptosis process and the cell will go for the cell death. And if the P53 check and halt and repair the DNA, then it will go for the cell entry into the cell cycle process. Additionally, PBT3 can regulate the expression of various genes responsible for DNA repair, cell cycle arrays, and apoptosis. If it properly repair that DNA, then it will forward the cell cycle. If the DNA is properly not repaired, then it will go for the apoptosis process. It can activate different genes that promote DNA repair, such as P21. This is protein 21. Its molecular weight is 21 kilodalton. So this is protein 21, which inhibits activity of CTK because CTK is the master regulator of the cell cycles and prevents cell cycle progression. It will inhibit the cyclin CTK complex, and if this cyclin CTK complex will be inhibited, then the cell cycle will not forwarded from G to S, S to G2 or G2 to N. So this is very important process. If we go to the different P53 gene functioning as tumor suppressor, this play a vital role in regulating cell cycle and halting the growth of malignant cells. When cell experience DNA damage, or stress signal or abnormalities in the cell cycle, the P53 gene is fast activated. Its activation leads to the production of the P53 protein, which acts as a good transcription factor for the synthesis of this P21 protein or P53 protein. This P53 protein then act as a major tumor suppressor gene, because if mutation occurs in the p53 gene, there is a chances of cancer will occur in that animal or person. This is the master regulator. And this its main function is to arrest the cell cycle, because if this is mutated, then error prone replication will occur and error prone cell will form, and this is called cancerous cell, uncontrolled cell growth. So the p53 main function is to arrest the cell cycle during the G1 phase, to check whether DNA mutation or any error prone mutation is occur in that DNA, and this enabling the DNA repair mechanism to rectify any damage before proceeding to the cell division. Then uh, 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 it will rectify the damaged DNA, or it will go for the cell death. If the damaged DNA will not repair properly, then the fate of that cell is for apoptosis or cell death. This is the major protein that is tumor suppressor gene. One is P53. Till the slide clear or not?
Yes, sir.
Till this, yeah. Okay. Then another topic is intracellular protein turnover. Then what is the intracellular protein turnover? This intracellular protein turnover is two types. One is ubiquitin proteasomal pathway. Another one is degradation of the different cytosolic proteins. If you go to the structure of this ubiquitin proteasomal pathway, this in the ubiquitin proteasomal pathway, the covalent binding of the multiple ubiquitin molecules. If you go to the structure of these ubiquitin molecules um, binds to the protein, marks them to for rapid turnover. Rapid turnover means degradation of that protein. If one ubiquitin will be tagged to a protein, the fate of the protein will be degraded. How this degradation will occur? In the ubiquitin will bind the enzyme E1. This enzyme E1 first activates ubiquitin by using one molecules of ATP, and this ubiquitin E1 complex is activated in ubiquitin enzyme E1 complex is activated. Then this activated ubiquitin is passed to a numerous ubiquitin conjugating enzyme. Another one enzyme is E1 is replaced by E2 ubiquitin activating enzyme, and this activated ubiquitin activating enzyme, then it will keep a transfer a particular ubiquitin specific target protein. Then this ubiquitin E2 and this E3 enzyme, this is called ligase. This means this will bind, this will help bind to the E2 ubiquitin to the target protein. If we see the structure, this green one is ubiquitin, and this is the E2 ubiquitin enzyme complex, and this E2 ubiquitin enzyme complex will bind to the target protein with the help of this E3 enzyme. This E3 enzyme is called as ligase. Ligase means it will ligate to the target protein. This ubiquitin is tagged is ligase to the target protein, and this target protein will bind. This is the target protein, and this target protein having a lysine residue, ubiquitin will bind to that lysine residues, and this lysine residues then polyubiquitination occurs after one tag, then another ubiquitin is ligate to the one molecules of ubiquitin. Then this ubiquitin polyubiquitin protein ligation tag This will be detected by proteasomal pathway or this is called proteasome. This in this proteasome there is a different types of peptidase enzymes are there. They will digest the protein molecules and release into peptides or amino acid fragments. And these peptide and amino acid fragment will be recycled. Okay.
Sir, can you
So
describe
what are the important?
some EON, please, again? I can't
Okay.
understand.
Okay. Uh, this First ubiquitin, this ubiquitin tag will be attached to the unfolded or targeted protein which having fate is to be degraded. First, the E1 enzyme will activate the ubiquitin by the help of one molecule of ATP, and this E1 enzyme activated these ubiquitin molecules because this tag is not activated, and this enzyme E1 in the help of a T molecule it will act this activated this ubiquitin molecules. After this E1 ubiquitin complex activated, then it will transfer the ubiquitin molecules to the E2 because E3 is a ligase and it will detect only E2 ubiquitin complex, not E1 ubiquitin complex. So ubiquitin is transferred to the E2 enzymes, and this E3, this is a ligase, and these ligase enzymes attach these ubiquitin molecules to the target protein, and this target after the attachment of the ubiquitin molecules to the target protein, the fate of that target protein will be degraded in the proteasome complex, and this proteasome complex having different enzymatic system that is peptidase which will keep the peptide bond to release peptides or amino acids. So fate of the um, target protein or unfolded protein or stress protein is degraded into peptide, and this will be recycled by the cell.
So
Understood?
what is the importance of this
Thank you, sir.
ubiquitin proteasome? What is importance? What are the important?
Big, okay. Uh, For an example, if you go to the college, what is the importance of your ID card?
Sir, for identification,
Very good. For identification, which protein will be degraded? How the proteasome will identify? The proteasome will be identify this ubiquitin tag. If one protein is tagged with ubiquitin molecules, then proteasome will detect that protein and degrade that protein. So ubiquitin tag is necessary because when the ubiquitin tag will be attached to the protein, when the protein is unfolded or properly not folded, or any stress condition like heat stress, cold stress, the protein is unfolded. So at that time, the fate of the protein is degraded. After tagging with ubiquitin, the fate of the protein will be degraded. This is understood. Okay.
yes, sir.
Then if we go to the second one, the of protein turnover, that is degradation of cytosolic protein. If we go to the degradation of cytosolic, when the cytosolic protein is degraded during your starvation condition or any stress condition, the protein fat will be degraded to utilize that amino acid to produce energy. At that time, only the protein turnover will occur. How this process will occur? During the cellular deprivation or starvation or any other stress condition, this can take up and break down specific cytosolic protein for its energy production. These targeted proteins under such situation carry the amino acid residues, particularly like here ubiquitin tag, here the protein having lysine, phenylalanine, glutamic acid, arginine and glutamine. If these sequence is there in the cytosolic proteins during the starvation period, this cytosolic protein fat will be degraded by the lysosome, and this is called autophagic process, which may route them to the lysosome for breakdown of lysosomal, because lysosome have different proteolytic enzymes, and this will degrade the different protease enzyme degrade the targeted protein, and these proteins are sacrificed to furnish amino acids and energy during starvation, enabling certain essential metabolic function to operate. Because during starvation condition, when the glucose is utilized, then after the glucose, the major source is the amino acid. Then body will degrade the amino acid, and during the this starvation condition, this degrade of this protein will occur by a particular signal, that is lysine, phenylalanine, glutamic acid, arginine and glutamine, and this signal will target that protein into the lysosome for degradation. Inside the lysosome, different proteolytic enzyme is there, and this proteolytic enzyme different proteins cleaves that protein to produce amino acid, and this amino acid will furnish our energy during starvation condition process for metabolic function to operate. Although ubiquitin is well known for directing protein into the proteasome or other processes, ubiquitination process by the help of this proteasome. Its participation in other cellular processes gradually being discovered, and its role in sorting target over by lysosome is envisioned, because maximum processes ubiquitin process is utilized, but during the starvation, deprivation or any stress condition, the lysosome fused with the organelles, and this process is called autophagic process. In this process, the different cell, also sometimes some cell organelles also fused with lysosome for our energy productions, or different types of enzyme, sorry, cytosolic proteins also fused with the lysosome to produce amino acid. From the amino acid, the energy will produce by different metabolic pathways, and this is called the protein turnover. And this protein turnover will occur by these two pathway. One is ubiquitin proteasomal pathway, and another is degradation of cytosolic proteins by lysosomes. These are two main pathways. Okay, till that slide clear?
Yes, sir.
Yes, sir.
Okay. Then the in the unit third, this is the last last one is animal cell culture. What is animal cell culture? Animal cell culture is a laboratory technique. This is a in vitro system. This is a laboratory technique used to grow and maintain animal cell outside their natural environment under control condition, and it is widely used in biotechnology, medical research and pharmaceutical development, because any animal cell will be cultured outside the lab, outside the animal cells, and this is in a laboratory condition to utilize, or any other in biotechnological field like vaccine production, or any research like cancer research, or new drug development, or any other medical research through this animal cell culture. If you go to the key requirements for the animal cell culture, because animal cell culture requires sterile environment, Because why sterile environment? Because our body is daily infected with multiple of bacteria or viruses, but our immune cell clears that viruses. But in laboratory condition, there is no immune system. So particularly this animal cell culture will occur in a sterile environment, and this will prevent the contamination from this bacteria, fungi, and different types of viruses. And next one is culture media. In animal cell, we use different nutrition supplements, but in culture media, same nutritional supplements is required for that animal cell culture. In that, amino acid, vitamins, different types of growth factor also are required. And these are the culture media, the DMEM, RPMA 1640, and PFBS. We'll discuss about this. Our third one is incubation condition, because our body is maintained in 37 degrees. Same in laboratory conditions, cells are maintained in 37 degree with 5% CO2 to mimic physiological condition. Why 5% CO2? Because in our body, blood used as good buffer system in our body. The bicarbonate buffer system is here. Here also we mimic that condition with adding some 5% CO2, and this CO2 will produce a bicarbonate buffer system to the cells, which is properly maintained during this incubation condition. And fourth one is aseptic techniques. Because aseptic technique is required during the cell culture or cell harvest or cell storage, use laminar flow hoods, because these laminar flow hoods used by different filters, like it will purify all the air during the sterile air condition. The cell culture will properly occur, and this all the equipment will be sterile, because the infection will be occur, because there is no immune system during this cell culture process. And sometimes we also add some antibiotics to the cell culture medium to avoid any contamination condition. Then after the cell grow, then we do passaging, that is called subculture. And in this subculture, transferring cell to a fresh media or continuous growth. Because during the suppose we keep the cell to different culture media, that is this supplement. After the supplement is over, how the cell will grow. Then we remove this supplement and add some fresh supplement by doing soft culturing or transferring to a new fresh medium, because we remove this medium and we'll add some fresh medium. Like we eat 3 times a day, like that, and the cell also we also feed to the cultured cells. Then, if we go to the different types of animal cell culture, then we'll go. First one is primary cell culture, that is derived directly from animal tissue like liver, skin, kidney. Directly pumps that say tissue, and it will digest with different tryptic enzyme, because they're uh, the cell will easily separated, and this maintains original tissue characteristics and limited lifespan, because they are finite cell division, because some our somatic cells having finite number of cell division because of the telomere sequence or haploid limits. And we can culture the in primary culture is the human fibroblast culture, and in secondary cell culture, that is derived from the primary culture by transferring cell to a new dish. Means we culture from this primary cell culture. Suppose blood cell will be taken, then we will segregate the different blood cells. Then after the segregate of the different blood cells like RBC, and other cell macrophages and neutrophils, then we will secondary cell subculture there. And then this can be passaged to multiple times. If multiple quantity cells are there, then will be this is called primary cell culture. And if we go to the third one, that is continuous or immortal cell line, like the cancerous cell. These are immortal in nature because, as we know, we discussed about P53 protein. If P53 is mutated, then the cell will undergo cancerous because no inhibition is there. The cell cycle is continuously occur. Here, if the P53 is damaged or mutated, then there is no inhibition of the cell cycle. Then the cell cycle will continuously process and it will produce numerous of the cells and it will cause a cancerous tissue, uncontrolled growth. There is no control in our cell. So these are immortal cell lines. And if you go to the subdifferent cervical cancer cell line, these are embryonic kidney cell line, these are Chinese hamster ovary cell line. Then we culture the cell. If we go to the fourth one, we culture the cell into two types, in a suspension and in a adherent culture. Why suspension? Because blood cells are transferred in a suspension manner, like in a liquid medium. But in our different side, like cervical cancer or kidney cancer, they are attached to the one surface because adhere. They are adhere into a surface. So in adherent culture, cell require attachment to the surface to grow, like fibroblast, epithelial cell. It required a surface for growth. If you go to the suspension of the cell line, like blood cell, sort of cancerous cell, blood cancerous cell, like leukemia, they require in a suspension or a liquid medium. They will grow in a liquid medium. And then, what is the difference between 2D and 3D cell culture? In 2D culture, traditional monolayer culture on a flat surface. Monolayer means single layer. If the layer after the single layer, if another layer will be occur in the monolayer, above the monolayer, then it will go for the 3D culture and it will produce like a tissue-like manner. So, this is called a 3D cell culture, and this is the basic of the animal cell culture. Then like in our body, some growth factor also required for the growth of the cell. Here also we also add some growth factors like growth media for animal cell culture. In growth media, we provide essential nutrient to support survival, proliferation, and function of the culture and animal cell. And these media contain a balanced mix of the amino acid, vitamins, salts, glucose, and different growth factor and antibiotics for inhibition of the contamination. If you go to the different culture medium, how we supplement to the cell, one is natural medium that is derived from biological fluids or extract or used in only research but less common now due to variability like plasma, serum, tissue extract, biological fluids, amniotic fluids. This extract will be added to the culture medium for growth of the cells. But maximum time we are not using this natural medium. We are using the synthetic medium or defined medium. This will mimic like natural medium. In this medium, chemically defined composition or controlled growth condition, more consistent and reproducible than natural medium used in research by technology drug development like DMEM, RPMA 1640, A12, and MEM. We also describe this. What is the difference between these two culture media? If we go to the key components of this culture media, then first one is amino acid, which is essential for protein synthesis. This is the important because this is the building block of the cells. And maximum time glutamine and arginine and leucine, these are essential. So these would be there in growth media. If it's second one is glucose because glucose produces energy for cell metabolism, so glucose is used in this medium. And salt electrolyte because electrolyte is balancing is required for osmosis, osmolarity maintenance, so maintaining osmotic balance and cell function. And vitamins also the essential required for metabolic reaction because these vitamins are the cofactors for enzymes. And the pH buffering agent sodium bicarbonate, because bicarbonate buffer system is required. Our blood is bicarbonate buffer system, so we may make to use sodium bicarbonate. And serum, that is fetal bovine serum APS, contain growth factors and hormones. These growth factors and hormones are also required for growth of the cells, and this hormone will give the signaling to the different cells. And antibiotics is optional, because if you know the cell is contaminated or will be contaminated, then we'll add some bacterial antibiotic, that is penicillin or streptomycin, to inhibit the uh, contamination, because in cell culture there is no immune system. Then, if you go to the different synthetic media like the albigous modified Eagles medium, that is DMEM, this is high glucose condition. Maximum time in cancerous cell, we use this high glucose condition medium, and because of the maximum times cancerous cell utilize maximum times glucose, they are using glycolysis process. They uh, in this process, the maximum use glucose molecules. This rich in also amino acid, vitamins, and glucose, and used for wide range of adherent, means attachment of all cells like fibroblast, endothelial cell, neuronal cells, or any breast cancer cell, colon cancer cell, like that. Uh, the next one is Russell Park Memorial Institute, that is RPMI 1640 synthetic medium. In this, the glutamine and high phosphate level is maintained, suitable for immune cell and lymphocyte and leukemia cell lines. Because phosphate and glutamine, these are essential amino acids, and phosphate is required for the DNA synthesis process. Here, the maximum time for we use this RPMI 1640, and these are the minimum essential medium. If we growth of cell, we maintain the cell slowly, then we use basic medium or minimal essential medium, often supplemented with fetal bovine serum for better cell growth. And another one is F12. This is Chinese hamster ovary cells and neuronal culture, often combined with DMEM to form DMEM-F12, which support the broad range of cells. Sometimes this other cell line, like without animal cell, other cell, like fish cell line or hamster cell line, like that, we use this combination DMEM and F12. Then scopes modified alveolus medium enriched in vitamins and amino acids, suitable rapidly for proliferating cells like cancerous cell, and hybridoma for production of different vaccines and hematopoietic, different hematopoietic stem cell. We use this IMDM synthetic medium. Then
Sir, what is F12? F12 harms
harms
F12. What is this? Uh,
this is extracted from the Chinese hamster ovary. Okay,
sir, what is the benefits of the cell culture?
because this extraction from this Chinese ovary, this is rich in the different types of growth factors.
Yes.
Okay, then we go for the which cell will culture, and which cell line will be cultured. And if we go to the different cell line, the cell line is a population of cells derived from a single cell and grown in vitro under controlled condition like laboratory condition. This cell can be used for research, drug development, and biotechnological application like cancerous cells. If we go to the cell lines, which cell will be cultured? Then cell lines are one is primate, finite or primary cell lines. This is derived directly from normal tissue, and this cell line having limited lifespan, because uh, can divide only for a few passages and maintain characteristics similar to the original tissue. And if your example is human fibroblast and a monkey kidney cell, these are finite cell, but Maximum time we use is continuous or immortal cell line, that is cancerous tissue, because maximum research are doing on cancer, because cancer is immortal. There is no uncontrolled growth. So maximum time this cancerous tissue are artificially immortal and can divide independently under proper condition, because uncontrolled. Then we used a large-scale production of vaccine development and cancer research. Uh, we do, because if we synthesize a new drug, then we'll first test on in vitro condition like this HeLa cells or breast cancer cell or embryonic kidney cancer cell or Chinese hamster ovary or any human lung carcinoma cells. We'll test that drug to this cell. If this drug will work on this, then we'll go for the mouse model system, because mouse model is similarity of gene similarity with us. So then after mouse model system, then we go for the pig or any other primates like monkey or chimpanzee. Then we go for the apply to the human. Then maximum time we commonly use cell lines and their application. If you go for the human cervical cancer, then cancer research virus study, because sometimes human papilloma virus also infected co-condition like cervical cancer in genetic research. If we go for the human embryonic kidney cell gene expression study, protein production, and Chinese hamster ovary for biopharmaceutical production, monoclonal antibody and vaccine will produce from this ovary, and human lung carcinoma, lung cancer research and drug screening, all the breast cancer drug screening, because maximum cancer immortal cell line is used for this drug research and drug screening, and only Chinese hamster and monkey, these are also used for only vaccine development. And in mouse fibroblast, cellular transformation study and cancer biology study also. Macrophages is a immune cell line, so immunology and inflammation research will be occurring in this macrophages cell line. If you go to the different characteristics of these cell lines, adherent versus suspension growth. Adherent grow attached to the surface, that is a epithelial, as we discussed. Suspension means in a liquid medium, that is like blood cells. Or normal versus transform. Normal cells maintain typical growth property, or transfer cell exhibit uncontrolled growth during cancer research maximum time. Or species specific difference. Human, mouse, hamster, and monkey cell line are commonly used for different research field, like I discussed.
Excuse me, sir.
Yeah,
Can I ask you one question, sir?
yes.
Sir, if any person suffering from cancer, so any
Hmm.
treatment happen, how she cure about the,
If any person is suffering from cancer, which stage of cancer? Because if firstly, sometimes people
for
are our.
example, brain cancer.
Suppose some lump will occur in our skin or under any our part of the body. Our cultural issue is there, so they will not discuss. After certain 4 to 5 years, they will discuss about that part, that something is here, one lump is here or swelling is here. Okay, after testing that, this person is suffering from type 2 or type 3 stages, different stages. So that is the major problem. If any swelling or lump is there, if we go for the analysis, that is cancerous or not, at that time, if we treat that person, the person should be recovered. But in different stage, like we discussed, this is a immortal and it will grow rapidly. If we see the structure of this, okay, here, if this P53 is mutated, this is a tumor suppressor gene. It will not suppress any tumor or cancer cell. Then the cell cycle is growing, growing, growing. One cell is produced 2, then G1 is G2, 2 cell is 4, then 4, 8, 16, 2 to the power n. And this
Yes,
will
yes.
form a cancerous in nature, uncontrolled. There is no control over there. So any drug will not respond. So that is the major challenge. Okay. So after the different cell line, how we maintain the different cell lines? We maintain the different cell line in growth medium, in cell culture medium, as we discussed different cell culture medium like DME, RPN 60, and we incubate that cell line in a temperature in a CO2 incubator, that is in temperature is 37 degree for mammalian cell, and CO2 concentration is 5% required for the bicarbonate buffer system to maintain the physiological pH. Humidity is 95% because humidity also required for the prevent evaporation of the culture medium, because in 37 degree if humidity is not there, then it will evaporate the all the medium and the cell will be dry. So this incubation condition will be maintained. After the cell is properly grow, then we go for the passaging or subculture. In the subculture process, we prevent the overgrowth of the maintain healthy cultures, and adherent cell require enzymatic detachment like trypsin. Or in suspension culture, then we are diluted into fresh media like adherent culture like kidney cells. In suspension culture like blood cells, we then go for the subculture. And major is we prevent the contamination using sterile techniques, biosafety cabinets and filter media, and regularly we also check the bacterial infection, fungal and mycoplasma contamination. Then if we have not any work, then we store the different cell line in liquid medium. Long-term storage is necessary to preserve different cell line for future use. Two primary methods maximum use, that is cryopreservation or short-term refrigeration. In cryopreservation, cells are frozen in minus 80 degrees Celsius with the help of DMSO, and then will transfer into the 196 liquid nitrogen. Then steps for cryopreservation process, the prepare cell, then harvest the cell in logarithmic or growth phase, because growth, if you transfer the growth phase cell, then during the harvest it will grow rapidly. If in a quiescent phase, if you store that cell, it will not grow rapidly. So growth phase is very important, and count the adjust the cell density, how much cell is stored, and prepare freezing medium maximum time cryoprotectant DMSO or glycerol
Sir,
DCO.
what is harvest cell, sir? I do not idea about.
Harvest means if you store the growth phase cells, then after the you harvest means then next time of you uh, put out the cell from the liquid nitrogen and culture in the uh, incubation period time or incubator or in laboratory condition. That is harvest.
Thank you.
Then freezing. How will store in the minus 180 or 196 condition, like we use cryoprotectant like DMSO and glycerol. This will protect the outer of the cells, and cell will not be hampered by ice crystal formation. Then slow freezing process will slowly freeze the cells from minus 1 to minus 8, then minus 8 to 82, sorry, 196 degrees Celsius. After storage, then after storage of the cell, when we require, then we remove one vial for culture, and in this process we call them thawing of cell, like uh, any vial you put in the fridge and you come out the fridge, it will be ice crystal. Then it will thaw it through your palm and rapidly thaw will in 37 degree, and all the cell will be harvested from that vial, and we remove the DMSO, and uh, then we continue the cell culture. Then
Sir, what is DMSO?
DMSO is a chemical we used for the protectant, cryoprotectant. It will not freeze the cells because cell having water molecules. So you
Sir,
know that cell having 70% water, and this 70% water will be ice crystal. and if we form high crystal, it will be brittle and a breakdown. So DMSO dimethyl sulfonate is a sulfoxide is a chemical which protect the outside of the cell and cells to not form ice crystal during the freezing process.
Yes, sir.
Yes.
Then we
Sir,
go for the cyto. Okay,
sir, can you please explain the thawing of cell again in simple words?
okay. After you store the cell in minus 196 degrees Celsius, then you remove from that 196 or minus 80 degree. Then you rapidly thaw that cell because from 196 it will take time to come to 37 degree. So if you thaw using your palm and thaw rapidly, it will fastly thaw the cells and the cells are coming in the medium. It form a solid like structure during the preservation at minus 196 degree. Okay. If we thaw that cell, thawing means what? Na, to come out from uh, minus 196 degree to 37 degree normal temperature. Like freezing, you put any water in your freeze at 4 degree or minus 2 degree or 1 degree, it will freeze the water. After you removal, it will take time
Yeah.
to reform to water. It will form ice. If you thaw that bottle, small bottle, it will fastly converted from ice to water. So thawing is required. Okay.
Okay, sir. Thank you.
Then after cell culture, how we use cytotoxicity study? If we synthesize one new drug to test on this cancerous cell and kill that drug is killed or not, we studied that process by cytotoxicity. Cytotoxicity means killing of that cell. Cytotoxicity refers to the ability of a substance or agent, such as chemical or drug, in any new drug synthesized, then we will put into the cell and the cell will dead or not will check. That process is called cytotoxicity, and this or different types of toxin on immune cell to damage or kill that cell. It is a key parameter in evaluating the safety and efficacy of drugs, chemicals, and environmental toxin in biological research and medicine. If we go to the different cytotoxic process, one is necrosis, that is uncontrolled passive cell death caused by severe stress or injury, like your pus formation at the womb area. That is called necrosis. If you put that drug, the drug is doing like that, necrotic death. This characterized by cell swelling, membrane rupture, and inflammation. But apoptosis is a programmed cell death, and that this triggered by internal or external signal. In this process, cell will shrink, chromatin will condense, and apoptotic body will form, and this apoptotic body will be taken by different macrophages or neutrophils for a proper cell death. In autophagic cell death, this is the self-degradative process where the cell digests its own components and it can act as a survival mechanism as we discussed in protein turnover or lead to cell death in extreme conditions. These are different types of cytotoxic condition. How we study this cytotoxic condition? If we treat a drug into the cell and we'll see which type of the cell death occur, either it is a necrotic or apoptotic or autophagic cell death. Then we'll study by different assay. One is MTT assay. This MTT, this is a metabolic activity via mitochondrial enzyme converting. If we treat a drug to the cell, if some cells are dead or some cells are live. Some live cell use this entity to form a formagen and we estimate that for formagen to know how many percentage of cell is killed or how many percentage of the cell live. This is like that. And another one is alarm or blue assay. This is the fluorescence based assay. detecting also metabolic activity, how many cells are metabolically active, and some non-toxic and allow real-time monitoring because these are real-time fluorescence dye and we will see the signal, fluoretic signal, so we know that real-time monitoring. And another one is trypan blue. This stains the dead cell blue while live cell remain unstained. The dead cell, because the dead cell membrane is fixed, then the trypan blue is attached to the membrane. Then it will stain that membrane to a blue-like manner, and live cell membrane is not permeable to this trypan blue, and this is selectively flow. As we know, live cells are selectively permeable. These dye will not enter into the cells, so the live cell will not stain. Only dead cells are stained. And different membrane integrity asset and detect cytotoxicity based on damage to the cell membrane by say, uh, you can use scanning electron microscope or uh, tran uh, or you can use scanning electron microscope to see the cell membrane integrity, either any pore formation for the cell that will occur or not. Or you can use lactate dehydrogenase assay. This LDH enzyme also released from the damaged cell, only damaged cell, indicate membrane disruption or necrotic like cell death. You can you can use this assay or any PI staining. This is propydium iodide staining. This is a fluorescence dye that enters only dead cell because dead cell only penetrate this dye. A live cell will not penetrate because live cell membrane is selectively permeable that cannot transport this dye. Only dead cell will enter the dye and it will give a red fluorescent signal, and we will see that how many cells are dead or how many cells are live. We can count them. Then we can use
Sir,
apoptotic.
previous slide one doubt.
Okay.
Uh, uh, Research alarmar blue assay.
This is
Again,
a
explain sir this.
This is a fluorescence-based assay detecting metabolic activity like enzymes. This metabolic enzyme will cleave this resurging blue to give a blue fluorescent signal. This will tell that how many cell gives this blue fluorescent signal, that is live cell, and which cell doesn't give this blue fluorescent like signal, and these cells are dead. We can count also them under the fluorescence microscope. Okay. Then we can use different apoptotic assay for programmed cell death. We also you can study molecular techniques like how apoptosis different apoptotic assays they are like Caspase-3 assay. The end of the apoptosis process is caspase-3 enzyme which degrades the cells, and if this enzyme will increase, then we know that the cell undergo apoptosis. If we go to the annexin B and PI staining, these also bind to the phosphatidylserine because of apoptotic cell, because phosphatidylserine is inside of the cells, and it after the cell death it flip to the outside of the cell, and this annexin B and PI bind to this phosphatidylserine and it will give a red signal, and we know that this cell is undergo apoptotic. Then we describe about the caspase of the apoptotic assay, and sometimes we use tunnel assay to detect DNA fragmentation because P53 detect the DNA fragmentation. If P53 is mutated, we cannot know the DNA is fragmented or not. Then we do the tunnel assay, and in tunnel assay, these compounds bind to uh, we can see that DNA is breaked or not, or any mutated or not by this tunnel assay. And sometimes also some reactive oxygen species. This is the oxidative stress induced cytotoxic assay, and a TCFS DF also gives the raw signal. And if a damaged cell produce reactive oxygen species, this is the stress signal which damage the membrane, lipid, protein, and DNA. And this assay know that the membrane is undergo apoptosis, and last one is glutathione. This is the main antioxidant acid, and this antioxidant told that this cell is or undergo oxidative stress on this glutathione sequester different types of stress releasing factors to maintain the balance between oxidant and antioxidant. And this assay will say that the cell is live or dead. This is the all about the different cell viability assay to study
Sir, all
in
the points, assay word is commonly used in all the points. It means why this word is repeatedly
assay means
right?
estimation.
Yes, yes, sir. Sir,
Okay.
kindly I request you the first slide. Can you explain again, sir?
First slide. Which one? This one. This one.
Yes, sir.
This is the different cell cycle process. This is the This is the growth of the cell cycle. This is the DNA replication of the cell, and this is preparation of for mitosis of the cell, and this is the division of the cell process. If you create a wound in your cell, how this cell will form or repair? This cell is repaired through this controlled manner, that is your cell cycle. First cell growth will take place. Then this cell growth having the DNA molecule is replicated and increase the amount, and then it will prepare undergo mitosis process, preparation for mitosis process, because for mitosis different proteins and enzymes are required. For this in G2 all the synthesis will take place. Then it will go for the mitotic division. In the mitotic division, from one cell it will give two, two to four, four to six, like that your womb will be filled. This is the main reason of the cell cycle. Okay, understood?
Sir, today and previous class PPT, PDF, can you share our WhatsApp group?
Nah, I share all the PPT and also the syllabus yesterday. Some people also today morning I have
Today class PPT, sir.
today I have seen uh, yesterday's PPT. Today's PPT I will send that time. Okay,
Sir, yesterday PPT in our WhatsApp group,
not there. Okay,
no, sir.
you just ping a message, then I will send to you. Then you will send your group. Okay.
ସାର୍ ଏଇ ଯୋଉ 4 ଦିନର ଇଏ ଅଛି ଏଗୁଡା ପୁରା ପଠେଇ ଦିଅନ୍ତୁ ସାର୍ ଟିକେ।
ଆଜି ତ 3 ଦିନ ହେଲା।
ହଁ ସାର୍ 3 ଦିନ ହେଲା, ଏ 3 ଦିନର ପୁରା କାଲି ଲାଷ୍ଟ ରେ ସାର୍ ସବୁଗୁଡାକ 4 ଡେଜ୍ ଯାକର ପଠେଇ ଦିଅନ୍ତୁ ସାର୍।
ଯଦି ସବୁ ଗ୍ରୁପ ର ଜଣେ ଜଣେ ମତେ ଟେକ୍ସଟ କରିଦବ କି ହ୍ୱାଟ୍ସଆପ କରିଦବ, ତାହେଲେ ମୁଁ ତମକୁ
ସାର୍
ସମସ୍ତଙ୍କୁ ପଠେଇଦେବି। ଆପଣଙ୍କ ଗ୍ରୁପ ରେ ଆପଣ ସେୟାର କରିଦେବେ। ଓକେ,
ଥ୍ୟାଙ୍କ ୟୁ ସାର୍,
ସାର୍
ଥ୍ୟାଙ୍କ ୟୁ ସାର୍।
ଆପଣଙ୍କ ନମ୍ବର ତ ଆମେ ଜାଣିନୁ।
ନମ୍ବର
ନମ୍ବର ତ ମୁଁ ସେୟାର କରିଥିଲି। ନମ୍ବର ତ ସେୟାର କରିଥିଲି, ବହୁତ ପିଲା ତ ମତେ ମେସେଜ କରିଥିଲେ, ମୁଁ ତାଙ୍କୁ ପଠେଇ ଦେଇଛି।
ଆମେ ଜାଣିପାରୁନୁ।
ହଁ ସାର୍ ତାହେଲେ ଆମେ
ଠିକ
ଠିକ ଅଛି
ଅଛି
ସାର୍,
ସାର୍।
ଠିକ ଅଛି ଆମେ ଇଏ
ମୁଁ
କରୁଛୁ ସାର୍,
ଆଉଥରେ
ଆମକୁ ପଠେଇ
ମୁଁ
ଦିଅନ୍ତୁ ସେଇଠି।
ମୁଁ ଆଉଥରେ ବି ମୋ ନମ୍ବର ଆପଣଙ୍କୁ ପିନ୍ କରିଦଉଛି, ଏଠି ଦେଖିଦିଅନ୍ତୁ ମେସେଜରେ।
ହଁ ହଁ, ୟେସ୍ ସାର୍।
ସାର୍
ଓକେ, ମତେ ଇଏ କରିଦିଅନ୍ତୁ, ମୁଁ ଆପଣଙ୍କୁ ପଠେଇଦେବି ଯାହା ବି ଅଛି। ଓକେ
ସାର୍ ଆପଣ ବଢିଆ
ସାର୍
ବୁଝଉଛନ୍ତି। ଆମେ ଆକ୍ଚୁଆଲି ଏଇ ସବୁ ବିଷୟରେ ଆଗରୁ ଟିକେ ଆଇଡିଆ ନାହିଁ, ତ ଆପଣ ବୁଝେଇବାରୁ ଟିକେ ଇଜି ଲାଗୁଛି। ପଢିବା ପାଇଁ ହେଲ୍ପଫୁଲ ହବ ବହୁତ।
ହଁ, ମୁଁ
ଥ୍ୟାଙ୍କ
ତ
ୟୁ ସାର୍।
ସେଥିପାଇଁ ହିଁ ଆପଣଙ୍କୁ ଏମିତି ବୁଝଉଛି ଯେମିତି ଆପଣ ଲେଖିପାରିବେ। Okay,
ଆଜ୍ଞା ସାର୍।
ସାର୍ ଗୋଟେ ଡାଉଟ୍ ଅଛି
Uncle?
ସାର୍ ଇନ୍ ଏମଟିଟିଏସ୍ ଆସେ
Hmm.
ଯୋଉଟା କହୁଥିଲେ
Hmm.
ତ
Hmm.
ଏମଟିଟି କନଭର୍ଟ ହଉଛି ଆମର
ହଁ ଫର୍ମାଜେନ୍
କୋଉଟା
ଡାଏକୁ
ଯୋଉଟା
କରିଦଉଛି।
ଫର୍ମାଜନ୍
ହଁ
ସେ ଫର୍ମାଜନ୍ ଟା କଣ?
ଫର୍ମାଜେନ୍ ଇଜ ଏ କ୍ରିଷ୍ଟାଲ ଲାଇକ୍ ଫର୍ମ ଫର୍ମାଜେନ୍ ଡାଏ। ଫର୍ମାଜେନ୍ ଆସେ କରିନ ତମେ +2 ରେ କ୍ରିଷ୍ଟାଲ ଲାଇକ୍ ଷ୍ଟ୍ରକ୍ଚର। ସେଇଟା ଗୋଟେ କଲର
କେମିଷ୍ଟ୍ରି ଲ୍ୟାବରେ
ଡେରିଭେଟିଭସ୍।
ହେଇଥିଲା କ୍ରିଷ୍ଟାଲ।
ହଁ କ୍ରିଷ୍ଟାଲ ଲାଇକ୍ ଷ୍ଟ୍ରକ୍ଚର ଫର୍ମ ହବ। ସେଇ ଯୋଉ କ୍ରିଷ୍ଟାଲ ଲାଇକ୍ ଷ୍ଟ୍ରକ୍ଚର ଫର୍ମ ହବ ସେଇଟା ଏନଜାଇମ୍ ଟା କନଭର୍ଟ କରେ ଏମଟିଟି କୁ ଏ ଫର୍ମାଜେନ୍ କମ୍ପାଉଣ୍ଡ ଆଣ୍ଡ ସେଇ ଫର୍ମାଜେନ୍ କମ୍ପାଉଣ୍ଡ କୁ ଆମେ ଡିଏମଏସ୍ ରେ ଡିଜଲ୍ଭ କରିକି ଆମେ ତାର ରିଡିଂ ନଉ ଯୋଉ କଲର ଫର୍ମେସନ ହେଲା ସେଇ ରିଡିଂ ରୁ ଆମେ ଜାଣୁ କେତେ ସେଲ୍ ମରିଛନ୍ତି ଆଉ କେତେ ସେଲ୍ ବଞ୍ଚିଛନ୍ତି। ଓକେ।
हेलो हेलो सर।
ଆଜ୍ଞା ସାର୍ ଧନ୍ୟବାଦ।
ହୁଁ।
सर apoptosis ଆଉ ସେ necrosis ର ଟିକେ example ଦେଇକି ଟିକେ explain କରିବେ ମାନେ
ସେଇଟା
କୋଉ
ଡିଟେଲ୍ସ
case?
ପଢିବା ଆପୋପ୍ଟୋସିସ୍ ନେକ୍ରୋସିସ୍ ଆମେ ଡିଟେଲ୍ସ ପଢିବା।
अच्छा
ଆପୋପ୍ଟୋସିସ୍ ଆଉ ନେକ୍ରୋସିସ୍। ଆପୋପ୍ଟୋସିସ୍ ମାନେ ପ୍ରୋଗ୍ରାମ ସେଲ୍ ଡେଥ୍। ନେକ୍ରୋସିସ୍ ମାନେ ଆପଣଙ୍କର ଯେତେବେଳେ ଉଣ୍ଡ ହଉଛି ଆପଣ ଦେଖିବେ ସେ ଯେତେବେଳେ ପସ୍ ଫର୍ମେସନ ହଉଛି ଦ୍ୟାଟ୍ ଇଜ ୟୋର ନେକ୍ରୋଟିକ୍ ଡେଥ୍। That cell is undergone necrosis. So that heal will be will not properly. But in apoptotic, if a wound is formed and properly filled, that is your apoptotic wound. But in necrotic, the wound is formation of pus will occur because maximum time bacterial infection is there to form a necrosis. If this treatment of different drugs, some cell are burst, they release all their hydrolytic enzymes, and this will affect the surrounding cells. That is called necrotic. But in apoptotic, they will form a small like vesicle manner, and that vesicle is engulfed by different macrophages or immune cells, and it will clear out easily. So this process is occur properly. This is your
सर सर
apoptotic.
जो ट्रांसफॉर्म सेल ସେଟା only restricted to human beings ନା other animals ରେ ବି ହୁଏ ନା ତାଙ୍କ ପାଖରେ କିଛି immune ଅଛି ସେ ट्रांसफॉर्म सेल କୁ ନେଇକି?
ट्रांसफॉर्म सेल पाखरे
ସେଟା କେବଳ ହ୍ୟୁମାନ ବିଙ୍ଗ ରେ ହୁଏ ନା ଅଦର ଆନିମଲ୍ସ ରେ ବି ସେଟା ହୁଏ ନା ତାଙ୍କ ପାଖରେ କିଛି ସ୍ପେଶାଲ
ନା
ମେକାନିଜମ
ନା
ଅଛି ସେ
ଆମେ ଯଦି in vitro condition, any in vitro condition, there is no immune system. In vivo condition, like in our body, there is a immune cells. But in vitro condition, there is no immune cells. So we'll put some antibiotics in that culture medium because infection will be not occur. If infection will occur, they will eat the cells. They will destroy the cells.
ओके सर
Okay.
ତାର କ୍ୟାନ୍ସର ସେଲଟା କଣ ପ୍ୟାରେଣ୍ଟାଲ
Cancer cell ଟା
କ୍ୟାନ୍ସର ହଉଛି କଣ ପ୍ୟାରେଣ୍ଟାଲ ଡିଜିଜ?
cancer is not a parental disease. If some parents have some mutation or any other problem, sometimes cancer will also arise,
ଓକେ
but not a parental disease because cancer is totally uncontrolled cell growth. Because when we eat in outside, we eat plenty of like different types of chemicals or any type of red color, yellow color, different color is additives is there. These additives mimics our different hormones. So these hormones, when body is not required, these hormones will produce, and this will function metabolically. They activated our cells, and this repeatedly activation of this cell causes the mutation in our DNA, and this mutation of this DNA or any error in their DNA will cause a in mutation DNA or any error DNA will produce a non-functional protein. If you go for an example, P53 gene is mutated, then it will produce a non-functional P53. That P53 will not bind for if you mutated P53 will not bind properly to this complex CTK inhibitor complex. So this cycle will continue and it will produce an uncontrolled like manner cell growth and this will cause cancer.
सर ମୁଁ ଏଇ क्वेश्चन ଟା କରୁଥିଲି कि जो अनकंट्रोल्ड ग्रोथ होची सेल रो जो कैंसर को
Hmm.
जंती हम ह्यूमन बीइंग रे सेटा
Hmm.
अदर एनिमल रे भी होची ना सेमानकर किछ स्पेशल मेकानिज्म अछि सेटा को सेमाने सेल्फ क्योर हे जाउछंती
ସେଲ୍ଫ କିଓର ହଉନାହାନ୍ତି ସେମାନେ ଆମ ଭଳିଆ ଖାଉନାହାନ୍ତି ଯାଇ ବାହାରେ କଲର ଫୁଡ ଯାଇ ଖାଉନାହାନ୍ତି
ମାନେ ପ୍ରୋसेस ବି ସେମାନେ ଖାଉନାହାନ୍ତି ବଟ୍
ଜଙ୍କ ଫୁଡ
ସେମାନ
ଖାଉନାହାନ୍ତି ହଁ ସେମାନଙ୍କୁ ବି ହଉଛି ବହୁତ କିନ୍ତୁ ବହୁତ କମ ଦେନ୍ ହ୍ୟୁମାନ ବିଙ୍ଗ କାରଣ ହ୍ୟୁମାନ ବିଙ୍ଗ ସୁପିରିୟର ସବୁ ଖାଇଛି ତ ଦ୍ୟାଟ
ତାର
ୱିଲ୍
ଓନ୍ଲି
କମ୍ପେୟାର
କରୁନି ଓନ୍ଲି ଲାଇଫଷ୍ଟାଇଲ ତ ମେଣ୍ଟେନ କରୁନି କେତେ ଜଣ ମାନେ ଭେଜିଟେରିଆନ
lives
ହେଇଛନ୍ତି
because
ସେମାନେ ବି
diabetic, cancer, and polycystic ovarian syndrome is a lifestyle disease.
ଓକେ
Nowadays, this is under lifestyle disease. Because our change in lifestyle, these disease will occur. All family having one diabetic patient, and all the girl student below the 16 to 23 undergone PCOS, polycystic ovarian syndrome. This is the major problem.
ଓକେ yes sir
Okay, these are the
Yes, sir.
major
Yes,
problems
sir.
due to
ସାର୍
this
ଆଉ ଗୋଟେ
lifestyle
କ୍ୱେଶ୍ଚନ ଥିଲା
factor.
ଆଉ ଗୋଟେ କ୍ୱେଶ୍ଚନ ଥିଲା ଯଦି ମନେକର ଜଣେ ମାନେ କ୍ୟାନ୍ସର ଡିଜିଜ ରେ ମାନେ ସେ ଆଫେକ୍ଟେଡ ଅଛି ଫିର ତାର ନେକ୍ସଟ ଅର୍ଗାନ ମାନେ ଗୋଟେ ଅର୍ଗାନ ରେ ତାର କ୍ୟାନ୍ସର ହେଇଛି ଫିର ତାର ନେକ୍ସଟ ଅର୍ଗାନ ବି ମାନେ ଆଫ୍ଟର ମାନେ ଟ୍ରିଟମେଣ୍ଟ ସେ କଣ କରୁଛନ୍ତି ନା ମାନେ ନେକ୍ସଟ ଅର୍ଗାନ ବି ତାର ମାନେ ଆଫେକ୍ଟେଡ ହଉଛି ତାହେଲେ ସେଟା ଟ୍ରିଟେବଲ
No, this is not treatable because that cell is undergone metastasis.
okay but the stage is 2 so is it treatable or
Sometimes, the stage is 2 is controllable, but if the cell is undergo metastasis, they will undergo different organ. Then they will cancerous at that organ, and the organ will be damaged, and the fate of the patient is to some month to some year.
डिस्टेंट
So
ऑर्गन को
whether
तो
it is genetic or not.
सर डिस्टेंट ऑर्गन को
Nowadays,
जदी
okay.
मतलब डिस्टेंट ऑर्गन को जदी मेटास्टेसिस करेल तो स्टेज 4 ही जाए ना
Distance organ को metastasis. If some person having a breast cancer, then it will affect the lungs or any or near the organ. Then it will damage rapidly. The patient fate will be dead.
So is it genetic?
Nowadays, this process is some genetic factors are causing this cancer because the lifestyle factors changes the some genetic factors. Due to the change in our DNA sequence.
sir what is Anchorage test
What is
Anchorage test
onco?
Riggs test
Some protein, oncogenic protein, like as we say, they like for the breast cancer, some tests as there called BRCA1, BRCA1, BRCA2. This marker protein tells that the person having breast cancer. These marker are called oncogenic test.
oh, okay okay thank you sir
so 2D cell culture used for which disease sir?
All the 2D cells are we used for cancer. For different treatment of different drugs to cancer cells. 2D cell and also we use 3D cells. Three dimensional cells means it formed from one cell form a tissue. That is a 3 dimensional cell.
Okay understood
Okay.
Thank you,
ओके
sir.
आज क्लास क्लोज करिबा मु शेयर कर देबी मुझे टेक्स्ट कर दियो शेयर कर देबी
Thank you, sir.
ओके
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you so
Thank
much.
you, sir.
Thank you.